United States Department of Agriculture.
All right. First, I just want to thank the organizers for allowing me to present on this topic. This title, Think Big, Not Small, to Reduce N overall. It's just a little saying that hopefully when we finish with this talk, you can understand where it comes from. This talk is fundamentally about reducing sample size. When people think about sample size, they often think about just the animals. And things that we need to remember is that animals will share big units. Those big units might be cages, tanks, or pens. Those big units might be an animal room, might be a lab, it might be a point in time. And things about these big units have variability among them. The big units will differ, and that matters. That matters. It can add variability, and how we deal with that variability is important. So since this is 3 R's, one thing that I will talk about is refining our statistical analyses. And what we want to do when we refine our statistical analyses is to account for those big units in our statistical analyses. When we do that beforehand, before we ever do the experiment, it turns out that we can reduce our overall sample size, which is just great. Okay, so first, refinement. A lot of animal researchers are not veterinarians, but they perform medical procedures on animals. And sometimes there might be a better way to do that medical procedure. And so they are asked, hey, why don't you refine your animal method to reduce the suffering on that animal? And the animal researchers they're game to learn that. They seem to be, yeah, I'll learn how to do that. Now, I know that this reduction is talking about animal numbers, but it also, if we refine our methods, we can reduce other things as well, like reduction of suffering in animals. Now, if we refine our statistical analyses to account for the big units, I've received some pushback from investigators like, oh, I'm not a statistician. I'm like, well, you're not a veterinarian either. So the things that we're talking about today, they're not terribly difficult. And if we learn how to refine our statistical analyses at accounting for these big units, we're going to be able to reduce statistical errors. If we refine our statistical analyses after the experiment has happened, we'll be able to reduce our statistical errors. If we think about things beforehand, before that experiment, we can reduce our overall N. So I'll be talking about refining our statistical analyses, but I'm not talking about any new statistical method here. In fact, the things that when I talk about accounting for big units, non-animal users use them already and have for decades. It doesn't seem to be as popular in biomedical animal research. And there are 2 experimental scenarios that I want to talk to y'all about today. Besides the overall message of refining your statistical analyses to account for the big unit, for these 2 common experimental scenarios, I want to give you 1 question and 1 fact that you can walk away with. The first experimental scenario is where we have same group cage mates, where you have group housing and all the animals in the cage are the same group. I want to give you a motivation of why it's important to ask, how are those animals caged? And then I'm going to show you some data and give you just some statistical facts that more cages and less animals is better than fewer cages and more animals per cage. The second experimental scenario I want to talk about is replicating for reproducibility. Sometimes folks will do an experiment and then do it a couple more times to show reproducibility. And so I want to motivate why it's important to ask, hey, how are you analyzing those replicates? And then give you some data and some statistical facts of why pooling the replicates is the better way to go. All right, so since I'm talking about power and things, just some baseline assumptions. The target statistical power that I'm working on here today is about 80%, maybe as high as 90%. But if it goes over 90%, well, then, that means that we can probably reduce our overall sample size. other, just a baseline assumption, I'm just going to assume that statistical significance is 0.05. That's the p-value is less than 0.05. I want to say it's statistically significant. That's great. And another thing is that I'm going to assume that all these hypotheses tests are two-sided. All right, so now to our first experimental scenario. Before I talk about same group cage mates, let me just talk about cage mates for a moment. Here, again, throughout this talk, the small units are going to be the animals. And in this situation, the big unit is the cage, right? So I've got two cages, the same group cage mates here. These animals, they're all of the same color, so they're all the same group. These animals are the same group, but these cages are going to differ. Well, sure, they're going to differ because they're different treatments, but they might also differ for other reasons, right? And here in this mixed group cage mates, in this one cage, I've got animals of different groups, so that's why I call it a mixed group cage mates. And these cages, they will also differ. They can also differ, right? So on that, when I talk about these cages differing, these big units differing, I'm looking at a cage rack. And I know these animal rooms, the environments are tightly controlled at 72 degrees Fahrenheit, at 60% relative humidity, 12 hours lights on, 12 hours light, tightly controlled. However, when investigators consider these 4 mice in their statistical analyses, they often think of these 4 mice as being totally independent of each other. But they're sharing the same cage, whether it's a mixed group cage mate or a same group cage mates. These 4 mice are sharing the same cage. And from 6 to 10 feet away, it looks pretty homogeneous to me, so I don't see what the problem is. Here's an example from agriculture. Here are corn plots. So I'm looking at these corn plots and, at 125 feet up in the air, these corn plots, they look pretty homogeneous with respect to the layout. Same size, same weather conditions, same light and dark cycles, everything. I mean, it's pretty homogeneous. However, an entomologist who goes in here and is wondering if these bioengineered corn are more resistant to a particular pest, when she samples 5 plants from that plot and statistically analyzes them, she knows that those 5 plants they're related. They come from the same plot. And when she analyzes them, she's going to take that into account. In education research, here's a second grade wing of a school. And at 300 feet in the air, these classrooms, they look a lot alike, right? And there's 20 second graders in this class. And, the mid-year standardized test, the scores in these 20 second graders are going to be more alike than the scores from these 20 second graders. And an educational researcher, you better bet they're going to take that class effect into account. Here's a subdivision. All these houses at 500 feet in the air, all these houses, they look pretty homogeneous. Same weather conditions, the internal temperatures are about the same size. But if an epidemiologist samples 2 biologically unrelated adults of similar age from this house, that epidemiologist, he's going to take that residence into account when they analyze it. So this intra-cluster correlation, attention needs to be paid to it. If we ignore these big units in our statistical analyses for the mixed group cage mates problem, when I've got in our big units, if there's different groups represented in our big unit, and I ignore that big unit in my statistical analysis, I open myself up for type 2 errors. My p-values are going to be artificially too high. They're going to be inflated. And that's here the big units are cages. On same group cage mates, if I ignore this big unit right here in my statistical analysis, I'm opening myself up for type 1 errors. My p-values are going to be artificially too low. And since this tends to be a more egregious error, I'm going to focus in on this. So I told you I want you to have a question that you can ask when you come up on a group housing experiment, whether it's before the experiment happens, you're reviewing a protocol, or a paper, you're reviewing a paper or reading a paper. Something you should ask is, hey, how are you caging those animals, right? Because how you cage them gives us an idea of what kind of statistical errors we're opening ourselves up to. Now, scientists, we like data. So I know that several of my collaborators said, is this intra-class correlation really a problem? Here's a real example. I've got 3 strains of mice. I've got 2 inbred strains, and the cross of them is C. And we're interested in comparing the behaviors of this hybrid to each of the 2 parental strains. We're going to compare behaviors. The animals were purchased. They're unrelated females they're not sibs. And they're placed into cages like this right here. I've got 8 animals from each strain, 4 animals per cage, same group cage mates. And this is repurposed data from an article by Wolfer and Company in 2004. It was a large experiment. They had 432 mice. I'm so glad that they presented their data with this article to make this work possible. So in their study, they had 3 labs, and each lab did this experiment 3 times. And they considered this experiment with no enrichment and enrichment. And so for each animal, they also collected 20 outcomes on each mouse. So that gives us 360 data sets. Now, on same group cage mates, before we look at the data, I need to talk about incorrect analysis and a correct analysis. So many people would use just an ANOVA for this same group cage mates problem. And when you do that, it assumes that these animals in the same cage are independent of each other, which may or may not be the case. Now, if we refine our statistical analyses and account for cage in our statistical model, that's what we should be doing. If this were a human study, for example, these are patients that are operated by one surgeon, and these are patients that undergo maybe a different surgery from a different surgeon, or maybe these big units are clinics, right? So in human studies, might be considered a cluster randomized trial, and their statistical methods worked out for that. Or it might be considered individually randomized group treatment trial, and there's ways to analyze these. And we can use those same methods for these biomedical animal experiments. I had a difficult time finding some articles on that. So when I asked to do this talk, I got after it and was able to get this paper submitted yesterday. The preprint is on Zenodo. And in this, I describe how to do statistical power and cost analyses and experiments when you've got same group cage mates. And in there, we account for the cages in the statistical model. So with that, let's look at the results. We're going to analyze it 2 ways. With the correct model, we can get estimates of that intra-cluster correlation. And when we do, 57% of those 360 data sets had non-zero ICC. That's statistically over half of those data sets had non-negligible ICC. Now, I said earlier that if we don't account for the big, we open ourselves up for artificially low p-values. So here are the p-values from the ANOVA analysis where we ignore the cage. And here's the p-values from plotted against the p-values of analysis that accounts for the cage. So if it's true, then we might see more p-values coming right here. Oh, excuse me. The smaller, ignoring the p-values, will be right here. And yeah, it is. We see that the p-values, when we ignore the cages, they tend to be lower. Now you see a whole bunch of them lined up on this line right here. It's for these is where the ICC was estimated to be 0. And so you get the same p-value. Now let's turn our example to a hypothetical example. We know that we should account for these cages, these big units on our statistical analyses. And if we refine our statistical analyses beforehand, then we might be able to reduce our overall N. So here's a hypothetical example. I got 40 mice. They're randomized between treatments A and B. I'm going to have 5 same group mice per cage, and that's going to give me 80% power to detect a 1.25 standard deviation difference between the 2 groups, 0.05 significance level test. And it does assume an ICC of 0.2, all right? Taking a cue from the human literature on cluster randomized trials and things like that, they say, hey, you know what? If you can, it's better to have more clusters, and it's okay if you don't have so many people in your clusters. Well, we're going to tweak we're going to tweak our experiment here, and we're going to increase the number of cages and spread our animals out among those cages. So I still have 40 animals, but now I'm going to have two same group cage mice, two same group mice per cage. And when I do that, I get 0.94 power. Everything else is the same. I just increase the number of cages and then spread out those animals among those cages and got more power. It's nice. So when we refine our statistical analyses to account for the cages beforehand, hey, we can reduce our overall N. We've got a lot more power that we've got more power of. So let's back off the number of cages we have. Turns out that 28 mice housed at two same group mice per cage, that'll give us 80% power. And we're able to reduce our overall N by 12 overall. That's a 30% reduction. So our question is, how are you caging your animals? And now the fact is, more cages with fewer animals is better than fewer cages with more animals. And we can see that with increased power and the ability to decrease our overall N. But that's not all. There's evidence that if we have fewer animals in a cage, there might be less aggression, at least among the male mice. Here's a paper from 2000, a really nice paper. And they present some nice data showing that, yeah, with fewer animals in a cage, the male mice are less aggressive. But there's more. If we put more cages with fewer animals in them, and say we forget to account for the cages in our statistical analyses and just use a regular old ANOVA, well, when we have more cages with fewer animals, it can reduce our type 1 errors. Here is, if I use the correct analysis, my type 1 error, when I use the correct analysis, is at 0.05, my significance level, right? And it doesn't matter what my intra-cluster correlation is. If I'm using the right analysis, my significance level, the type 1 error rate is going to be the same as my significance level. But if I use the wrong analysis and I have 5 animals per cage, well, what that type 1 error is going to increase as that correlation increases. If I have 4 animals, that type 1 error rate goes down a little bit, 3 animals, 2 animals. So if I inadvertently use the incorrect statistical analysis, but I have more cages with fewer animals per cage, it can reduce my type 1 error. That's when I'm using the incorrect statistical analysis. Hopefully we'll refine and account for those cages in our analyses. Now, when I tell researchers, hey, maybe if you spread your animals out, you'll use more cages, but you can use less animals, and that's great. And I said, Reid, the cage cost, it eats it up. Well, let's look at that for a minute. Let's compare the cost between two power equivalent designs. Remember, I had 40 mice in 8 cages. That gave me 80% power. And 28 mice in 14 cages. That gave me 80% power. Let's look at that. Suppose a mouse costs $25. That's all in cost. That's how much the mouse costs to buy and all the time that you spent into this mouse. It costs $25. That's not a very expensive mouse. And the cage per diem is $1 a day. At my institution, it's about $1.25, $1.25 a day. But yeah, suppose it's this. At 30 days, the 2 mice per cage is going to be cheaper than the 5 mice per cage. Yeah, it's got more cages, but it's only for 30 days. So it's going to be cheaper because you save on that overall animal cost right here. However, if you let that experiment run long enough, eventually the cage costs are going to overwhelm the overall animal costs, and the 5 mice per cage, 40 mice total, will cost less than the 28 mice total. And we could compute that time point at 51 days. And we see that the 5 mice per cage, 40 mice total is going to be cheaper at 51 days and following. So again, a benefit from more cages with fewer animals is that it may be cheaper to do it that way. So let's sum up on these same group cage mates scenario. When animals are group housed, they're going to become related. And if you have same group cage mates, you're opening yourself up for type 1 errors. It means your p-values are going to be too low. And if you have mixed group cage mates, well, then you're opening yourself up for type 2 errors or p-values that are too high. So we should be asking them, hey, how are you caging your animals? We can ask them when we're reviewing protocols, and we can ask this when reviewing papers. And when we're reading papers, we can think, well, what am I opening myself up for? Because they haven't told me, I don't know. So that's a question we can ask. And then the fact that I want to drive home is that more cages and fewer animals tends to be better than fewer cages and more animals. It's because you get more power, potentially less aggression in some species. You better foolproof yourself against errors if you do the incorrect statistical analysis, and it could possibly cost less, which is great. So that's our summary for the first scenario of same group cage mates. Now I want to talk about replicating for reproducibility. Sometimes people will do an experiment and they plan to do it a couple of more times maybe to show reproducibility. And here's a hypothetical justification I might see. And I read this, it says 22 rats were equally randomized between two non-survival surgeries, A and B, and it's going to have 80% power to detect a 1.25 standard deviation difference between the groups with a 0.05 significance level test. And I go, yeah, that sounds right. And I'm not worried about the caging because these animals, they undergo the experimental procedures at the end of life. So I'm not going to be worried about asking them about how are you caging these animals after the experimental procedures began. But then I see this. Hey, we're going to replicate this 2 more times for a total of 66 animals. I see 66 animals? Well, how are you going to analyze those replicates? That's what I'm going to ask. So let's talk about that for a second. These replicates, yeah, they're going to be different. I mean, they're different in time. And it's possible that the lab personnel change. You get new grad students or new lab personnel come in and out. The batches of animals will be different. The resources, the reagents and things that you use, it might be different batches. Things to think about, right? They're going to differ, but that's okay. In fact, those differences, that gives the experiment some external validity. But the similarities on these replicates were conducted with the same objective in mind. And they're conducted with the same protocol. And when they report results, they're going to report them in the same paper. So these replicates, that's part of the overall study design. So we should ask them, how are you going to analyze those replicates? Whether we do that on a protocol before it's approved or reviewing a paper, how are you analyzing your replicates? Because we definitely want to avoid something like this. I've seen this several times in high impact journal. Data are representative of 3 independent experiments. What does representative really mean? I'm going to ask them, hey, how are you analyzing those replicates? Now, if this was a human study, this would look very much like a multi-site randomized clinical trial. And you know what the principal investigator is going to do with data from these different clinics? They're going to analyze them together. They're going to pool the data. The statistical methods for doing that have been around for a long time. Here's a paper from '86 by Dr. Fleiss. He describes how to analyze those data from multi-clinic trials, how to pool the data, a correct way to pool the data that accounts for the big units here, and of course the incorrect way, which is ignoring the big units. And the method that he describes here is totally appropriate for the rat experiment I'm talking about. But the animal researchers, yeah, they're reluctant to pool these data because they're so different. They can be so different, they say. We want to remember that inter-cluster correlation. And when we account for that ICC, it's okay. But if we ignore it, yeah, this variability among these replicates, it's going to show up in our data. And when we ignore it, our p-values are going to be inflated. So what we want to do is refine our statistical analyses instead of using an ANOVA here, and an ANOVA here, and an ANOVA here, or maybe an ANOVA for all 3, which assumes that these animals are totally independent of each other. we want to refine our analyses to account for these replicates in our statistical model. I pointed to that article by Dr. Fleiss in the mid-'80s. There's a recent article by Fromlet and Hines where they propose a slightly different model to analyze different replicates. In that, they propose it's a good idea. I noticed that if they make a little tweak on how they compute their error degrees of freedom, you can increase the power and prevent some other statistical errors. So there's ways to do this, and they're not terribly complicated, but people want to see data. So let's look at it. Here's that same experiment again. The same experiment I showed you a minute ago. Now remember, there were 3 labs, and each lab did this experiment 3 times, right? Or these are replicated across time. Or if I just think about the first replicate, I've got each lab, they're replicated across space, right? I've got 3 labs conducting this first replicate. And again, we've got 2 enrichments and 20 outcomes on each mouse. So we've got 360 data sets. I said that, hey, look, don't appropriately account for that replicate. Well, your p-values are going to be too big. So I analyze it ignoring the replicate, analyze it accounting for that replicate. Let's see what the results are. Yeah, the p-values are kind of inflated. So pooling replicates, yeah, it's better. When we pool our replicates, we're going to get fewer false negative results. We'll increase our power, which is always good. So let's look back at our original justification. They said, hey, 22 rats is going to give me 80% power, but I'm really doing 66 animals. Okay, well, let's update that to account for those replicates in the analysis. And when we do that, I've got 22 rats and 3 independent replicates for a total of 66 animals. And now I have a power of just about 1. That's a lot of power. Why don't we back off that animal number a little bit? So when we account for those replicates in our statistical analysis beforehand, we can reduce our overall animal numbers. We do that here. We say, I've got 8 rats and 3 independent replicates, gives me for a total of 24 animals, and I've got 80% power. Nice. So we've accounted for the replicate. We've spread out our animals among these replicates, and we've reduced our overall N by 42 overall. So it was about a third of the total originally asked for 26 animals. So we're just a little bit more than a third of the original with 24 animals overall. So when we pool our replicates appropriately, it's better than analyzing these replicates independently or not accounting for the replicates in there, and we can decrease our overall N. So now let me wrap up. Scenario replicating for reproducibility. The question that we want to ask people is, how are you analyzing those replicates? Just because the replicates differ, that's not a bad thing. It can add external validity to your experiment if you account for them appropriately. And also those replicates, that's part of the study design. You've planned that beforehand. Go ahead and include that in your statistical analysis plan, right? And so when we do that, when we pool our replicates appropriately, account for them in the statistical analysis, we're going to have more power on our analyses, and given us more power on analysis, if we account for it before we do the experiment, we can reduce our overall sample size. So that's the fact that I want you to walk away with here, is that pooling replicates, it's better, especially when you account for that replicate in your statistical analysis. Okay, so there, I've covered the 2 experimental scenarios, and now I'll just give you an overall summary. You know, these small units, which are animals, they're often contained in big units like cages and tanks, maybe animal rooms, maybe different labs, different points in time. And so these animals that share a big unit, they're related. And as time goes on, this intra-cluster correlation can develop. And when we ignore those big units in our statistical analyses, well, if in my big unit, if all the animals in that big unit are the same group, ignoring that big unit in my statistical analysis is going to give me p-values that are too low. And animals in my big unit, like replicates, are mixed groups, and I ignore that replicate, or I ignore that big unit, my p-values are going to be too high. So refinement, animal researchers, they're game to learn new animal methods. It reduces animal suffering. That's great. Let's encourage them to go ahead and refine their statistical analyses and account for those big units in their statistical analyses. If they do that after the experiment, that's still great. It's better use of the animal's data. It'll reduce the statistical errors. That's good. And if they account for these big units before they ever conduct the experiment, well, then they can reduce their overall N. So that brings me back to my talk's title. If we think big, not small, we can reduce N overall. So that's what I wanted to tell you guys today. I'm grateful for Eric and Rofe there in my department, and they listen to me talk about this stuff and help me think through things. Yeah, I'm grateful to have them to talk with in my department. Also, Carrie Roberto, she's our AWIC administrator. She's given me a lot of information, a lot of perspective, animal users' perspectives, giving me good feedback on things. This work has been partially funded by these grants right here. So that concludes my talk.
USDA is an equal opportunity provider, employer, and lender.